Good morning, everyone, and thank you for coming here today. My name is Sumaya, and our team consists of 5 members who are as follows: Amira, Rinat, Aram, Jarud, and I. We are students at the University of Jeddah. The topic of our presentation is about electrophoresis. We will talk about some general information about electrophoresis, process of work, some extraction method, components, applications, and how to measure concentrations. We choose this topic especially because of the interest that we share of how electrophoresis is important for our specialty. And now I will present our team and the task allotted to each one of them. First, I will define electrophoresis, and later on I will narrative the application of electrophoresis with Amira, Rinat, and Aram. Also, I will sum up. In the second part of the presentation, Amira will explain the process work for electrophoresis. In the third part of the presentation, Rinat will talk about electrophoresis RNA, and Aram will explain nucleic acid extraction techniques. In the fourth part of the presentation, Rod will talk about electrophoresis component, and later on Rod will explain the method to measure the concentration of electrophoresis. If you have any questions, please hold them until the end of our presentation, and we will be very delighted to answer each one of them. And now, give us your attention, please, to introduce our topic. Now, I will define electrophoresis. Electrophoresis is an essential technology for the separation and analysis of nucleic acids. Electrophoresis of nucleic acid is used routinely at the lab bench for the isolation and manipulation of cold and DNA fragments. In addition, it is a critical component of many molecular biology protocols that assess the role and interaction of nucleic acids in cells and tissues. Nucleic acid electrophoresis has attained particular importance in the current era of genome sequencing. In this application, speed and accuracy of nucleic acid analysis are of vital scientific and economic interest. And the practice of nucleic acid electrophoresis has evolved to reflect these needs. Gel electrophoresis is useful in forensics, biochemistry, genetics, microbiology, and other applications requiring analysis of nucleic acid and proteins molecule, size and characteristics. Protein electrophoresis is also a common method for analyzing blood plasma in medical applications. Electrophoresis often occurs as a proprietary technique superior to calling DNA sequencing, sorting plotting, restriction fragment length, polymorphism, and analysis via mass spectrometry.
Now I will talk about electrophoresis, how the process works. Before electrophoresis begins, a user prepares and cools a liquid gel, puts into a tray, and feeds with the toothed comb to create separation for each individual sample. A buffer fluid containing conductive ion is then added to the solid fluid gel in order to sustain current flow and maintain contained pH. The user injects a dyed nucleic acid or protein sample into the gel, and the power supply passes an electric current through it. Larger and the longer molecule move slowly between the negative and the positive supply poles, while similar, shorter molecules move more quickly. Differently sized molecules therefore from distant the band in the gel, which they are analyzed or compared to the acid chain.
Edvotech instructional videos, performing electrophoresis. In this video, we're going to perform electrophoresis in 5 easy steps. Watch our video preparing the agarose gel tray before starting this experiment. Step 1. Place the gel tray into the electrophoresis chamber. Completely submerge the gel with diluted electrophoresis buffer. Refer to Table B for recommended volumes. Step 2. Load 35 to 38 microliters of the dye samples into wells and consecutive order, starting with the closest well. Step 3. Place the safety cover and check that the gel is properly oriented. Remember, the dye samples will migrate toward the positive electrode. Step 4. Connect leads to the power source and perform electrophoresis. Refer to Table C for time and voltage guidelines. Here's a time-lapse of our experiment samples migrating. Step 5. After electrophoresis is complete, remove the gel and casting tray from the electrophoresis chamber and visualize the results. No staining is necessary. And here you see the results.
Electrophoresis of RNA. Accurate determination of the size of RNA species is just as important as detection of the molecular weight of any other macromolecule subjected to electrophoresis. This is accomplished by comparing the electrophoresis of an RNA sample against molecular weight standards. The successful electrophoresis of RNA is accomplished in two parts. RNA is denatured prior to loading the gel, and during the electrophoresis, conditions that support and maintain the denatured state are established. These steps will ensure that like species of RNA comigrate, and the best possible resolution is achieved when gels are poured thinly and run at low voltage. The most commonly used RNA denaturant for agarose gel electrophoresis is formaldehyde.
Nucleic acid extraction techniques. Electrophoresis is used to determine the quality of the extracted nucleic acids. DNA is a negatively charged molecule and will therefore migrate towards the anode in the presence of an electric field in an electrolyte solution. Differential navigation determined by size. The electrophoresis of nucleic acids is usually done at slightly alkaline pH. This ionizes all the phosphate groups in the backbone of the molecule. Fluorescent affinity dyes, such as lithium bromide or SYPR green 2, are used to visualize the prepared sample. Electrophoresis allows the clinical laboratory to assess the presence of high molecular weight DNA after extraction. For smaller pure products, denaturating polyacylamide gel, electrophoresis will provide the best DNA length resolution, whereas non-conductive agarose gel are more appropriate to determine sizing of larger products.
Electrophoresis component. Electrophoresis components are often sold and recorded separately. Common equipment includes dyes, trays, power supplies, electrodes, cables, gel mixtures, and gel dryers. And chemicals such as denaturing agent, gel hardenant, influents. Selection of an appropriation gel is the most important to the electrophoresis process. Gels are layered into an upper stacking gel and lower separating resolving gel. The stacking layer is low in acrylamide and has low pH, while the separating layer has a higher acrylamide concentration and higher pH. The difference between the two layers results in high resolution and more distant bands for easier measurements. Three types of gel are used in the electrophoresis process: agarose gel, polyacrylamide gel, starch gel. Aqueous gel have a low resolution but a high range of separation. They are employed in DNA fragment analysis. Polyacrylamide gel are used for protein and small fragment DNA analysis. Starch gel are formed from partially hydrolyzed potato starch. They are slightly more opaque than aqueous or polyacrylamide types and are used for protein electrophoresis.
And now I will talk with Amira, Rinat, and Aram about the applications of electrophoresis. Antibiotic testing. Antibiotics help medical patients fight off illness and infections. Electrophoresis is used to separate the antibodies in the antibiotic from any impurities. This process also enables researchers to determine the concentrations of the antibiotic, making dosage more accurate.
DNA analysis. DNA analysis is one of the most common applications for electrophoresis. Using gel as a medium, researcher can stratify DNA into the segment using an electrical charge and keep the molecule in the place once the charge is removed.
Vaccine testing. Vaccines have saved countless lives and radically reduced the spread of diseases. Researchers use electrophoresis to test variations of vaccines with different levels and types of antibodies to conduct studies to find the best possible version of a single vaccine.
4. Analysis of proteins and antibodies. Electrophoresis that used for this application is called immunoelectrophoresis. Research can test samples from medical patients using immunoelectrophoresis to detect a wide variety of immunoconditions. Research can analyze the way that various antibodies interact with abnormal protein present in these samples to find potential treatment or even cures for autoimmune disease.
Electrophoresis method to measure the concentration. If we consider that there is hydrodynamic fraction on the moving particle due to the viscosity of this percent, then in the case of a small Reynolds number and medium electric field strength E, the velocity of the scattered particle phi is simply proportional to applied field, thus knowing the phase transition. Phi equal velocity, E equal field strength, and phase transition. Now phase transition equal velocity per field strength.
We have reached the end of our small presentation about electrophoresis. So, in conclusion, electrophoresis is a key analytical technique for the separation and analysis of protein, DNA, carbohydrates, and other biomolecules. It is an essential prerequisite for many techniques that are used in molecular biology and alongside chromatography and spectroscopy. Is a fundamental procedure in the laboratory. All in all, we have talked in this presentation about the most important information about electrophoresis through explaining about general information, process of work, some extractions method, components, applications, and how to measure concentrations. So, at last, we hope that this small humble presentation will benefit you. As well, we hope that this presentation will contribute to the delivery of information to your mind easily and that you will like it.